So just looking at this amazing pictures in this book, can you tell me what are mermaids? Who were the Amazons? Who were the Incas? Do elephants ever forget? Are elephants afraid of mice? What is this book?
This book is called Still More Tell Me Why. It was one of a series of books, Tell Me Why, More Tell Me Why, and Still More Tell Me Why. And it was really a science book that I would have spent a lot of time reading as a kid growing up, somewhere around between 10 and 12. I'd find a quiet corner. I can still see where I was in the house and hope nobody would find me. And I would read for hours.
Were you in a big family? Was there a lot of you?
I was second of, well, I was 4 at that time when by the time I'd reached 12, there was a 5th, but when I was reading this book, it was probably second of 4.
So there was a lot of activity in the
There was other activity in the house, yeah. And definitely there was jobs that probably needed to be done that I was hiding from. But it was, I would literally, find time, love to kind of get stuck into it and just read. And it was just one of those books that really fascinated me. And in particular, there were parts of it, the ones I always tended to veer towards were descriptions on disease and how disease is caused, and so there are some really. I would have been always up at this end of the book.
Okay.
With
That's funny. Like, did you have people in your family who were ill or who had diseases, or had you come across it in some other way? Or
no, I was just fascinated by it. Fascinated by what caused disease. I would have had no. There would have been nothing that would have triggered it from that point of view.
Yeah.
But just had a real interest in. Not just the disease, but the other simple things like why do humans have hair? When do you stop growing? Things. One of the things that's in it, I won't be able to find the pages. What is ESP?
yeah. And what is ESP?
Well, I think it doesn't exist. But would have been a thing. I think around the 70s in terms of extrasensory perception.
So is that like telepathy?
I think that's what ESP referred to. I've forgotten.
I was actually watching something on YouTube, so it must be true, last night, where somebody said that scientists have discovered that telepathy exists.
Well, then maybe it does. I must reread that part of it.
Yeah. What causes dandruff? Okay.
Yeah.
What are birthmarks? Oh yeah, I remember asking my father. I have a birthmark on my leg. I should have took the death, but I remember asking him, why do I have that? I was really upset about it, and he told me it's because if there was another girl in the world who looked exactly like you in every way and you got lost, we would be able to find you because you'd be the only one with that birthmark. Exactly. I was perfectly satisfied with that.
Brilliant answer. Yeah, brilliant answer.
Delighted.
Very good.
Yeah. So they're the kind of answers that you were finding in these books to these. And did you have the questions yourself anyway, or were you just...
No, I would just sit down and just find a page and just read. I was just fascinated. It used to totally absorb me.
Amazing.
So it was and there were other books like on the planets and so on, and which I'd have read, but not have had as much of an interest in as this. Why do we sing? Do people walk in their sleep? Why do people walk in their sleep? Love that stuff.
Yeah, absolutely. So, and you kept it, obviously, the book.
Well, yeah, it would have been in my parents' house. So when one says there was a clear up being done, and I was like, I want this one. This comes to my house. So
and can I ask you, when you saw that again for the first time, was it the pictures you remembered more, or was it the?
It was the feeling of just being totally absorbed. There was the pictures, yes, but it was just that feeling of being, this is, it can real kind of comfort of being, in my happy place.
Okay, brilliant. So can you introduce yourself then?
So my name is Siobhan McClean. I'm a biochemist, and I'm in the School of Biomolecular Biomedical Science in UCD. And I work on cystic fibrosis infections and also vaccines, or trying to develop vaccines for bacterial infections.
Well, that's kind of topical, isn't it? Or it certainly has been, this word vaccine. We've all heard the word vaccine, I'd say, in the last 2 years more than we've ever heard it before.
Yeah, there's never been a better time to work on vaccines. It's been very interesting. But so we don't work on viral vaccines, but we do work on bacterial infections and trying to get vaccines to protect against those. But it's certainly, there's never been a better time to work on vaccines. It's something that everybody kind of has some idea about anyway.
And what exactly is it that a vaccine, the kind of, I don't want you to go into enormous detail obviously, but like the underlying sort of idea of how a vaccine actually, what is a vaccine?
A vaccine, to put kind of simply, it's kind of training the immune system or telling the immune system this is something that you need to protect the body against. So it's like exposing the immune system to a portion of this
whatever
thing that's going to cause the infection, and so that when the immune system sees it again, it goes, aha, I've seen this before, and then is able to has the tools to mount a response to protect the body from that before the infection actually takes hold. So it's usually you're trying to develop a vaccine to prevent the infection, but often actually we'll try and get a vaccine which the person might still get the infection, but it won't be as bad. So it's kind of the immune system kind of kicks in and
modulates it.
So it doesn't do its worst.
It's not naive. It's not seeing it for the first time.
Yeah.
And do we have a couple of sort of very sciencey looking things on the table, but these are not particularly sciencey. Are these actually things that you would use in the lab?
Yeah, so this is what we call a gel in the lab. It's a little bit old, which is why it's got this beautiful curly edges. But this is how we look at proteins in the lab. So when we're trying to develop a vaccine, the vaccines that we develop are proteins. So they're molecules in the body that do work, that actually have effect in the body or in the cell. And so what we do is we look at it. We'll pick. See if you can see this this big band here. I don't know if that's visible on the video.
Maybe it'll be easier on this one.
So one of those, what we call a band, will represent a vaccine antigen. So in other words, it's the molecule that we're going to want to use as the vaccine, which will again stimulate this immune response and protect people. So what we do is we look at for particular molecules in bacteria, these antigens or vaccine antigens in bacteria, and we use those as we try and develop those as vaccines.
And you make that sound so easy. So once you found the molecule that you want to use, how do you actually then turn that into a vaccine?
So firstly, I suppose that we figure out what these molecules are, because they're the molecules that the bacteria use to actually stick to human cells. So that's our first step in the process. We try and find the molecules that bacteria use to attach to human cells. And then once we have those, we have to go back to the genome of the bacteria, take the gene for that protein, put that gene in another cell so that other cell makes lots and lots of the protein. Then we purify the protein, and we then, when we have the protein in a pure form, we test it, and we test it in mice. So we'll take groups of mice and we'll give the mice 3 of these immunizations, and in the same way as people got three COVID-19 vaccines. And then we'll come along, we'll give the mice the infection, and we work out if the vaccine works, the won't get the infection, and if the vaccine doesn't work, the mice will get the infection, and so that project will stop, and we won't go any further with it.
It's reasonably straightforward.
It's a reasonably straightforward process, from start to finish. It's once we know what the protein is, we have to figure out we have to get the gene and then transfer the gene and purify. Now it takes a long time.
We've described it there in the space of 3 minutes.
Yeah,
obviously,
it takes a long time.
A lot more complicated than that, and you've made it sound very simple, which I know it isn't. And the reason that, so that's probably why the syringe is here. The reason that you test it on mice, I can imagine people will say that's not right and we can't be doing that and all the rest of it. But the reason for that is...
Well, we can't test on humans.
Exactly.
So you have to test it. So the mice is actually a very well understood immune system. And so we can test them on mice, and we know if it works in mice, it has a chance that it'll work in humans. Often things work better in mice than they do in humans. So even if it works in mice, doesn't necessarily mean it'll work in humans, but it's the best chance we have of knowing before we test in humans. So we have actually one vaccine that we're hoping to actually test in humans in the next year. I'm hoping. Cross my fingers, and it'll be a very small trial that we'll do in maybe 10 to 12 people, and it'll be very much looking at the safety of it, because it has to be safe in people. So we start very, very small with only about 10 people. So I know a lot of people wouldn't be uncomfortable with the fact that we have to test things in mice, but there's no other way of knowing if it works without actually testing it in an animal, because you have to actually have the whole living mammal with a liver, with lungs, with the various organs.
Exactly.
Yeah, so that's the only way we know it works.
Yeah, so you can't just test it on a particular set of cells, because the particular set of cells, might. That's not how the immune system works. The immune system works as being part of.
You have to have a spleen, you have to have a thymus, you have to have a bone marrow, you have to have all those components. Yeah,
the immune system is really complicated.
It's complicated, but it's fascinating.
Yeah.
It is. Yeah.
I found it really, really hard when I was studying cell biology to understand. So the other thing I want to ask you about is this: is you hardly use this in your science, do you?
No, definitely not. That's kind of there because I love to get out up the hills. So I just love to get out walking, and I like to go up.
Really?
So if on a summer's evening, I'll head up to Braddon or Ticknock on a quick run. I say run, but it'll be walking up and down. And then if over a weekend we'll head up the mountains, and we'll go the Knockmill Downs, the Galties, the Mourns. We'll just take on a weekend and go walking. So it's great. It's lovely to be just out in the wilds, looking at what's there.
And you don't find actually climbing uphill hard work?
No, it's not. I mean, you take your time. You don't fly up. But no, it's not. It's lovely. And if you're with people, it's a great way to chat because you're kind of walking and chatting. Or if you're on your own, it's just a great way to be in your way with your thoughts. So it's a lovely way of spending time. So that's why that stick is there. I have very bad knees from bad knees. My knees are not my strongest. So the sticks are great for coming down.
Actually, I climbed. My cousin was over from America recently, and she dragged me off to climb a mountain, and I was amazed at how easily I got up. But I found coming back down really much more difficult.
Yeah.
I'm not a fan of hauling myself up mountains. I'd walk from here to Timbuktu on the flat.
Yeah.
I don't find it really hard to,
keep going. But the views are amazing when you get there.
Yeah, that's true.
The views are fabulous. But coming down, it can be tougher coming down. So and because I say I've done my knees skiing and things like that. So
you're obviously really active.
I like to be busy, but I'm not very sporty. But I do like to get out and about.
So the vaccine, Siobhán, that you said you were developing, is that what is that actually for?
So the one that we're hoping to bring to humans very soon is for a disease. It's a tropical infection called melioidosis, which is an infection that's very, very common in Thailand and Northern Australia, but actually spreading right throughout the tropics. And so if we get to humans and it works and it's safe in humans, that would be just amazing, because it really is it's a huge problem in, say, Thailand, where up to 40% of people die when they get the infection. It's
40%?
Really 40%, in parts of Thailand that get it will die from it, and it's partly unheard of in Ireland.
Never heard of it myself.
Yeah. So
and how does it affect people?
It causes it can cause septicemia, it causes liver abscesses, it causes these abscesses in their here, the parotids.
And it's a bacteria?
It's a bacterial infection, yeah. It gets inside cells, and once it gets inside cells, then it's able to hide from antibiotics, and it keeps...
I find it terrifying. I find all this microbiology and absolutely terrifying.
Yeah, but
these tiny single-celled or tiny viruses, is a virus even a living thing, and they can cause much, like, pain, suffering and death and pandemonium.
Absolutely.
And chaos and, economic. Sure, look at the COVID-19 vaccine, the whole world is going to its knees by a thing you can't even see.
But then the other side of that is within 2 years of the COVID-19 vaccine, COVID-19 coming, we actually had a vaccine. So yes, it can be frightening, but in fact we're getting better and better at targeting these things and tackling these things, I think.
And are bacteria more difficult to deal with than viruses, or there isn't a difference?
I think there isn't a difference. It depends on what they throw at you. So there's such a variety in all the bacteria that are there, and such a variety in the viruses that are there. So it just depends on what they throw at you. Yeah. But the reason we got into this tropical disease, people ask, why am I looking at a tropical disease that's based in Thailand and Northern Australia? And the reason is that we started the process by looking at a particular bug that causes infections in people with cystic fibrosis, which is so the one that the melioidosis is caused by a Burkholderia pseudomallii. The one that causes infection in people with cystic fibrosis, Burkholderia capacia complex, all big names. So we firstly, actually about 10 years ago, found a vaccine that works in Burkholderia capacia complex, because for about 10 years before that we were studying how Burkholderia attaches to lung cells and how this particular infection only affects people with cystic fibrosis, or predominantly affects people with cystic fibrosis. It doesn't affect other people. And so that was something that was really, really interesting, is how these bugs cause such a problem for people in CF.
Yeah, I wonder why that is.
So there's a few reasons. People with cystic fibrosis, their immune response wouldn't be as strong as people without CF. But
And why
also
is
they
that?
have...
Sorry to keep asking you questions.
No, it's fine.
Because the immune system isn't necessarily the thing in cystic fibrosis that's affected.
No, so they have the main genetic problem is a pump in their lungs, a chloride channel in their lungs that should be pumping out chloride, and that doesn't work effectively. So there's a build-up of mucus. There should be a layer of water out on the cells outside the lungs, and that gets dried out, and then the mucus builds up, and that mucus traps bacteria. But also the surface of your cells has little hairs that move like that to move bacteria up and out of the lung, and those hairs in people with cystic fibrosis don't work that effectively, and so bacteria then it's an environment that bacteria can thrive in.
Right.
So as a result, so for about 10 years, as I say, we were studying how bacteria affected people with cystic fibrosis and didn't affect others, and that's how we identified this vaccine that ultimately is going to have probably more impact in Thailand and Northern Australia than it would have in the cystic fibrosis population. They're two very closely related bacteria.
And if you could develop this vaccine for this one that affects people with cystic fibrosis, it would have a huge impact for them, wouldn't it?
This infection, back in the 90s, this infection in people with CF was quite prominent. About 30% of people with CF would have got this infection. In more recent years, because people with cystic fibrosis now have to be segregated from each other, they don't mix socially to prevent cross infection. This particular infection isn't as prominent as it used to be. So only 3% of people now with cystic fibrosis will get this infection. But that means that actually has huge impact on their lives because they can't mix with their peers.
Is that because of the pandemic?
That's the risk. No, this has been since the mid-90s. People with cystic fibrosis don't tend to mix with other people with cystic fibrosis, so they avoid infection. Yeah, it does impact on their quality of life. Wow. Yeah. It's probably one of the toughest things, that it certainly has an impact. But because of that, this particular infection isn't as common as it used to be, which is good because it's a very nasty infection.
Okay. It's a kind of a useful thing you're doing.
Well, it's fascinating for me as a scientist, but I would like to think that it will someday have real impact.
I hope so too. I'm sure it will.